The last component of this flow of energy is the cellular oxidation of glucose. And so once again, I'd like you to look at a module prepared for Biology 101 in which we look at the cellular oxidation of glucose in great detail. Today we're going to complete our discussion of energetics, and I need to tie up a few loose ends on photosynthesis, and then we'll get into the cellular oxidation of glucose. Now, starting with photosynthesis, one of the things I wanted to mention previously, which I did not, is that in your lecture outline, you have essentially a diagram that is really a study guide for you. So what you want to do in these processes is really tell the story of photosynthesis, or tell the story of the cellular oxidation of glucose. So I know you can't see the details of this diagram, but at least off of this camera. But again, you should talk about the light reaction of photosynthesis, flow of electrons, pumping of hydrogens, production of ATP, NADPH is the terminal electron acceptor, how those materials are used in the Calvin cycle, and so on. So again, this is a diagram to help you work your way through these processes, but it's not really a teaching tool I can use in this format. All right, now another thing I wanted to talk about is that when we do look at the light reaction of photosynthesis, there's something called the cyclic flow of electrons, and I didn't really get to that last time. So again, in the light reaction, of course, we're going to have flow of electrons, water being the source of electrons, NADP picking up electrons, reduced to NADPH at the end. So flow of electrons, pumping of hydrogens, production of ATP. Now, NADPH and ATP, of course, are used in the Calvin cycle or dark reaction to produce the glucose. But what NADPH can also do is that it can also deliver electrons back to chlorophyll A's that lose their electrons. So some electrons don't just go through the cycle once, they'll actually go through the cycle several times. And this is variable from one species of plant to another, and so we refer to this as cyclic flow. So generally speaking, 2 hydrogens generates a single ATP, and generally 2 electrons pump 2 hydrogens. So it's hard to put a one-to-one format on this for all species of plants, because some plants can get more ATPs per electron than other plants can. So that's certainly something that is interesting about plants and variable from one plant species to another. Okay, another thing I want to go back to is CO2 getting into the plants. I mentioned last time that the stomata, when they're fully hydrated, or the guard cells are fully hydrated, that opens the stoma, allowing CO2 into the leaf. And when they're dehydrated, that closes the stomata, preventing CO2 from getting into the leaf. And part of that is, again, in response to saving water. So what plants do is that they'll actually pump sodium into the guard cells in the mornings. That then makes the cell hypertonic, draws water into the guard cell, that opens the stoma. Now, as the day progresses then, this pumping of sodium can't overcome the dehydration of the plant, and so eventually if the plant dehydrates, the stoma will close. Now, I also talked about the fact that some plants are C4 plants and have this C4 pathway to concentrate carbon dioxide and get carbon dioxide in rapidly. And then another group of plants called CAM plants, crassulation acid metabolism plants, that also have a C4 pathway, but they alter the stomata control in a way that the stomata are open at night and closed in the day, further conserving water. So part of what these C4 plants and CAM plants are about is getting CO2 in quickly, preventing water loss. But another aspect of this is that it helps maintain CO2 levels within the cells. And the reason that that is important is that ribisco is a very inefficient enzyme. If we go back to the dark reaction, as I mentioned before, a typical enzyme will carry out about 30,000 reactions per second. Ribisco only carries out 2 to 3 reactions per second. Also, it will bind oxygen if it's available as over carbon dioxide. And the problem with that is that if ribisco binds oxygen instead of CO2, it triggers this process known as photorespiration. And in photorespiration, what it leads to is the overall degradation or loss of ribulose bisphosphates. ATP is consumed in photorespiration, and CO2 is released in photorespiration. So these are all counterproductive to photosynthesis. And so again, what these C4 plants and CAM plants are trying to do then is to maintain higher levels of CO2 in the cells to prevent photorespiration. Now, the other thing I wanted to get back to is also when we look at the dark reaction, the product is glucose. Why make glucose? What's the point of making glucose? Well, glucose, of course, is an organic molecule. So what the plants are doing is bringing in carbon dioxide, an inorganic molecule. They're bringing in water, combining the hydrogens, carbons, and oxygens in a way to make an organic molecule. And once glucose is produced in, other organic molecules can be made from that. So lipids, non-essential amino acids, and other organic molecules. So that's one value. But the other value, of course, is that glucose has energy value as well. So the glucose can be deconstructed then to generate ATPs. And so if we think about the overall flow of energy, you might be saying, well, why do plants need to do that? They have light. They can convert light energy to ATP. They have all the ATP they need from the light reaction. Well, don't forget it's a light reaction, light-dependent reaction. So what happens to cells that are in the roots? What happens at night? Well, the plant is still going to need ATP because ATP is needed by enzymes to carry out chemical reactions. So that means they're going to have to go back to some of their glucose, break it down to generate ATPs to carry out their metabolic processes when there is no light. So they will consume some of their organic molecules. What they don't consume then goes into their tissues, and we as consumers take advantage of that. So plants then not only have chloroplasts, they have mitochondria, and this is why they are able to metabolize their own sugars. All right, so let's go ahead then and get into the cellular oxidation of glucose. This is how we consumers then generate our ATPs. And again, ATP is critical because if enzymes have work to do, they have to get that energy from ATPs. So what we do is take in glucose, we combine it with oxygen, and that yields carbon dioxide and water. Also, it's an exergonic reaction. So 686 kilocalories of energy is released. About 263 kilocalories of that will be tied up in ATPs. So if this is a completely efficient process, we'll have about 36 ATPs, sometimes 38 ATPs. And also heat is released in this process as well. So most of it is released as heat. Some of it is trapped in the energy of ATPs. Okay, now the cellular oxidation of glucose occurs in the cytoplasm of the cell, the liquid portion outside the mitochondria, and also within the mitochondria. And mitochondria have a structure similar to chloroplasts in that there's an outer membrane and an inner membrane. The inner membrane is highly folded. These folds are known as cristae. And then there's a fluid-filled matrix in the center where a lot of enzymes are found. So when we look at the cellular oxidation of glucose, there are also sub-steps to this, just as there are in photosynthesis. So one of these is glycolysis, which occurs in the cytoplasm. And then cellular respiration is the part of this that occurs within mitochondria. And there are three sub-stages there: the conversion of pyruvate to acetyl coenzyme A, the Krebs cycle, and the electron transport system and chemiosmotic phosphorylation, also known as oxidative phosphorylation that we find in the mitochondria. Also, we're looking for a maximum of 36 ATPs per glucose. So again, this is just kind of an overview of where we're headed. We'll come back to this later, but glycolysis is going to yield 2 ATPs per glucose. Cellular respiration will yield 34 ATPs per glucose, none in the conversion of pyruvate to acetyl-CoA, 2 in the Krebs cycle, and the big payoff is going to be in the electron transport system, chemiosmotic phosphorylation. So that's where most of them come from. So what is important to understand is that cellular respiration occurs within mitochondria. And so cells that lack mitochondria cannot produce these ATPs. They can't produce these 34. They're dependent only on 2 ATPs per glucose. So prokaryotic cells lack mitochondria. They only get 2 ATPs per glucose. We eukaryotes, if oxygen is present, can get 36 ATPs per glucose. Okay, so let's go ahead and look at glycolysis. Kind of a quick overview is that we're going to start with a single glucose, and this is an enzymatic pathway. So we're going to start with glucose. We're going to end up with different products. And glucose then will be split into 2 3-carbon molecules, glyceraldehyde 3-phosphates. And we've heard that term before, or that molecule before in photosynthesis. And what's interesting here is that it requires actually the input of ATP. So we're actually going to consume ATP in these early steps of glycolysis, 2 ATPs per glucose. Then these glyceraldehyde 3-phosphates undergo another series of reactions, and eventually stay as 3 carbon molecules, although there will be some other materials attached to them, and these are known as pyruvates. So we're going to start with 1 glucose, we're going to end up with 2 pyruvates at the end of glycolysis. But in the second series of reactions, you'll notice that NAD is reduced to NADH, and that's an important step because it comes before the energy producing steps where ADP is converted to ATP. So we will have 4 ATPs produced then in glycolysis, 2 ATPs invested. And so when we look at the overall energy gain then, 2 ATPs are invested, 4 ATPs are produced per glucose, so a net gain of 2 ATPs. And then also 2 NADHs are generated, and these have potential energy value because, again, this is an electron carrier. It's going to deliver energy from those electrons at some point. Okay, now, glycolysis occurs in the cytoplasm outside of the mitochondria. And I also want to show you a video prepared by our publisher, which is actually really good at showing some of this stuff that we're interested in. So, in the cytoplasm then we have the glucose. And the glucose is going to be altered and ATPs are consumed. So you notice that 2 ATPs were consumed. It's going to be split into 2 3-carbon molecules. NAD then is going to be reduced to form NADH. And then this will be exploited for a substrate level phosphorylation. So ATPs are going to be generated through a substrate level phosphorylation. So 2 per pyruvate, which eventually would be 4 per glucose. So 2 ATP invested, 4 ATP generated, a net gain of 2 ATP. Now, the pyruvates then diffuse through the outer membrane of the mitochondrion, through the inner membrane of the mitochondrion into the matrix of the mitochondrion. And this is the next phase, the conversion of pyruvate to acetyl coenzyme A. And so in this phase, what happens, again, we'll do a quick overview and then look at an animation, is that pyruvate, a 3-carbon molecule, an enzyme will bind that, clip off one of the carbons, and it will be released as carbon dioxide. And NAD is going to be reduced in NADH, and then the remaining 2 carbons, which are described as an acetyl group, will be attached to something called a coenzyme A to form an acetyl coenzyme A molecule. And we'll find out its role in a second. So again, each pyruvate then has 1 carbon clipped off, the remaining 2 carbons, called an acetyl group, attached to a coenzyme A molecule, forming acetyl coenzyme A. So let's go ahead and see what that looks like then in our animation. So here's our pyruvate. We're in the cytoplasm. In the background here we see our mitochondrion. And so it's going to diffuse through the outer membrane first. So through the outer membrane of the mitochondrion, through the inner membrane of the mitochondrion, into the matrix of the mitochondrion. And so here's our pyruvate. So one of the carbons is clipped off, it's released as carbon dioxide. And one of the things you should know is that once carbon dioxide is produced, it's lost. We can't attach carbon back to organic molecules or carbon dioxides back to organic molecules. That's what the plants do for us. And then this 2-carbon unit called an acetyl group, an enzyme will reduce an NAD there, NADH is produced, and it will attach that to the coenzyme A. So here's the coenzyme A, here's our acetyl group, so it's an acetyl coenzyme A. Now, that brings us then to the next phase, and that's the Krebs cycle. So the role of the acetyl coenzyme A is to introduce those 2 carbons, that acetyl group, to what we call the Krebs cycle. And so the Krebs cycle, being an enzymatic cycle, again, we need to start and end with the same material. So we want to start with oxaloacetate. You can see that we're going to go through a series of reactions, and we're going to end up with oxaloacetate. And we're going to be bringing in 2 carbons, and we're going to be taking out 2 carbons. But I'll come back to that in a second. But what I want to focus on right now is really this area right here. We want to see the relationship between the acetyl coenzyme A, a 4-carbon molecule called oxaloacetate, and then citrate, which is a 6-carbon molecule. So what happens here is an enzyme will bind oxaloacetate, this 4-carbon molecule. it will bind the acetyl coenzyme A. And what that enzyme will do then is transfer these two carbons from the acetyl coenzyme A to the oxaloacetate. And when it does that, we now have a 6-carbon molecule with these 2 carbons attached to it. So we've got the 4 carbons with the 2 attached. So this is now a different molecule. It's called citrate. The coenzyme A is released, and the coenzyme A then can go get more acetyl groups. It can pick up more acetyl groups. So at the end of glycolysis, we have 2 pyruvates. That means that you'll have 2 acetyl coenzyme A's produced, which means that 2 oxaloacetates will be converted to 2 citrates. And again, the coenzyme A can be reused over and over again. So again, I don't expect you to see the details on this, but the general idea then is that oxaloacetate is picking up 2 carbons from acetyl coenzyme A, forming citrate. That'll go through a series of reactions, regenerating oxaloacetate. Now I'm going to show you a simplified view of this cycle then. So acetyl coenzyme A shuttling in 2 carbons, those being bound to oxaloacetate to form citrate. And then again, those 2 carbons come in, and one of those carbons goes out here as carbon dioxide, the other goes out here as carbon dioxide. And along the way then, NADs are reduced to NADH. And also FAD is reduced to FADH2. And we also have another substrate level phosphorylation. ATP is generated at one point in this Krebs cycle as well. So since we start with 1 glucose, that'll generate 2 acetyl coenzyme A's. So that really means we get 2 turns of this cycle. So 2 ATPs are generated by the Krebs cycle for each glucose that we start with. Okay, and let's go ahead and look at the animation now to look at these steps. So the acetyl coenzyme A then, and one of the weaknesses of this animation is that it doesn't show us the enzymes. So here is our oxaloacetate. So we're going to have a transfer of the 2 carbons to the oxaloacetate. So that forms citrate. And then an enzyme will bind that, remove the carbon. And also reduction of NAD to NADH. Another carbon dioxide is generated. Reduction of NAD to NADH. And this 4-carbon molecule will go through some other processes. ATP is generated at one point. We have reduction of FAD to FADH2. And of course, NADH being generated as well. Then at this point then, we've regenerated oxaloacetate, and acetyl coenzyme A can bring in 2 more carbons and start the cycle again. So that's going to bring us then to the big payoff. But I want to quickly come back to where we are in terms of ATP production at this point. So we're looking for 36 ATPs. 2 ATP per glucose are generated through glycolysis. We're looking for 34 in cellular respiration. We didn't get any from the conversion of pyruvate to acetyl coenzyme A. 2 per glucose through Krebs cycle. So the big payoff is going to be coming in the electron transport system and chemiosmotic phosphorylation. So let's go ahead and look at that animation. And this is one we've seen before, really. So the electron transport system of the mitochondria, we're looking at the inner membrane of the mitochondrium. This would be the matrix here, outer compartment here. And so the source of electrons are going to be the NADHs and FADH2s. So they're dropping off the electrons and they're passing through. And of course, as they go, they're pumping hydrogens into the outer compartment. So source of electrons, NADH, FADH2. Now, terminal electron acceptor, and so this is critical, is oxygen. So oxygen is going to be our terminal electron acceptor, and we'll work our way over there in a second. Here's the passing of electrons, pumping of hydrogens. And now we're getting to the terminal electron acceptor. So oxygen molecules will bind electrons and free hydrogen protons, and that forms water. And we refer to that as metabolic water because it's water produced in this metabolic process. And that just becomes part of the solution of the cell. Of course, the hydrogens are being pumped. They concentrate in the outer compartment and will eventually diffuse back through the ATP synthase, producing ATPs. All right, so as we look then at the value of the Krebs cycle, the value of the Krebs cycle isn't so much how much ATP is produced there, but it does produce a lot of NADH and FADH2. Then they become the source of electrons, which get pumped, and of course that leads to ATP production. So when we look at the source of electrons, some of them come from glycolysis, 2 NADH per glucose in glycolysis, 2 NADH per glucose in the conversion of pyruvate to acetyl coenzyme A, and then 6 NADH from the Krebs cycle, 2 FADH2 from the Krebs cycle. All right, and so again, the big payoff then comes in the electron transport system. Now, one question then is, why do we die without oxygen? I think everybody from a childhood knows that if you don't breathe, if you don't get oxygen, you're going to die. So let's now look at that in detail. Let's look at the step-by-step consequences of not getting oxygen, because there's really a domino effect here at work. So oxygen is the terminal electron acceptor. And so I'm going to go to, again, a diagram in your lecture outline. And I know you can't see the details here, but what we're looking at here is glycolysis. We're looking at electron transport here, Krebs cycle here, conversion of pyruvate to acetyl coenzyme A. So if there's no oxygen to be the terminal electron acceptor, at the end of the electron transport system, we will have potentially a source of electrons, but no terminal electron acceptor. So that's going to shut down flow of electrons. Electrons won't be able to flow. Without the flow of electrons, hydrogens cannot be pumped. If hydrogens cannot be pumped, we will not have production of ATP. So the problem is that once you stop the flow of electrons, you lose 32 ATP per glucose in the electron transport system. Now, if flow of electrons stops in electron transport, NADHs and FADH2s cannot drop off their electrons. So that leads to a shortage of NAD and FAD. So without NAD and FAD, the Krebs cycle shuts down. And so you just lost 2 more ATP. Now, without the Krebs cycle, and also with a shortage of NAD, you cannot have conversion of pyruvate to acetyl coenzyme A. So pyruvate builds up then in the cytoplasm. It will no longer diffuse into the cytoplasm. That creates now another problem in that the cell now will try to save itself. And so there is a part of glycolysis which is called anaerobic glycolysis or fermentation. And so as we know, glucose will form pyruvates. And pyruvate in an oxygen-deprived situation then in mammals can be converted to lactate. And this is an important step because what happens in this conversion to lactate is we don't lose any carbons, first of all. And secondly, you'll notice that NADH can be reformed to NAD. That's an important step because NAD is required before the energy producing pathways in glycolysis. So this is an important step. Now what's good about this though is that if oxygen is made available, lactate will be converted back to pyruvate and we can eventually get our 32 or 34 ATPs per glucose out of them in the mitochondria. So this is a reversible reaction, and this is what happens in our own cells, mammal cells. Now, if you were happen to be a fungus or a prokaryotic cell, you might carry out what's known as alcohol fermentation or ethanol fermentation. And here the pyruvates are converted to ethanol or some other alcohol. But the problem is that the carbons are clipped off. Carbon dioxide is produced. So this is not reversible. So again, they'll produce NAD. That NAD will get you 2 ATPs per glucose. But regardless of whether you're producing ethanol or lactate, those 2 ATPs aren't going to keep you alive. So that's a last-ditch effort to keep you alive. So again, as we look at the consequences of no oxygen, no terminal electron acceptor then shuts down the flow of electrons. That means no pumping of hydrogens. That means no chemiosmotic phosphorylation, so you lose 32 ATP. A shortage of NAD shuts down both the Krebs cycle and conversion of acetyl coenzyme A. Pyruvate to acetyl coenzyme A. Pyruvate builds up in the cytoplasm. You'll convert some to lactate to try to keep you alive for a while, but it won't be enough. And if you stay in an oxygen-deprived situation, the cell dies. Enough cells die, you die. Now, the last thing I want to get to then is just a quick summary of where the ATPs come from. So again, glycolysis will yield 2 ATPs per glucose. Cellular respiration, 34 ATPs per glucose. We don't get any from the conversion of pyruvate to acetyl coenzyme A. Krebs cycle will give us 2 per glucose. But again, the big delivery is electron transport, chemiosmotic phosphorylation, 32 ATP per glucose. So we have to have the oxygen to get that big delivery because we can't live on 2 ATPs per glucose. Now, the other thing briefly I want to get to is the relationship of our different foodstuffs. We've talked about the fact that proteins, carbohydrates, and lipids can all generate energy. They all have caloric value. And we've talked in detail now about carbohydrate metabolism, how carbohydrates eventually can lead to acetyl coenzyme A. That's the entryway to the Krebs cycle and the big payoff in ATPs. What I haven't really told you is that all of these reactions are reversible. So if you have acetyl coenzyme A's, you can put those carbons together to regenerate glucoses and eventually complex carbohydrates as well. Now notice that proteins can be disassembled to amino acids and again eventually to acetyl coenzyme A and again entry to the Krebs cycle and delivery in ATPs as well. Now you can't store amino acids though. Amino acids, once you take the amino group off, that goes to the production of urine. So the liver will produce urea that goes out in the urine. But those carbons have energy value. And again, the same thing with lipids. Lipids can be converted to acetyl coenzyme A, and we can get ATPs out of that. However, that also unifies all of these things. So a lot of people think, well, if I eat proteins, for example, well, I won't gain weight. And again, a lot of students a lot of times will be trying to build up, and they'll say, hey, Mr. Sourbeer, I'm getting fat. I don't get it. Well, what's happening is that if you can't build proteins with those amino acids, you're going to disassemble them down to acetyl coenzyme A's. And those acetyl coenzyme A's then the carbons can be put together to produce fatty acids or even glycerol, and you'll store that as fats. Same thing if you're eating lots of carbohydrates. If you don't need to produce ATP, you'll just back that up and produce fats from that. So that's the concept here. It is really all about calories. It's how many calories are you taking in and how many calories are you expending. So if you're not expending as many calories as you're taking in, even though you may not be eating fat, because all of these processes are unified by acetyl coenzyme A, you'll just build those fats and store that for later. So we really obviously require energy. Energy is a big part of the flow of energy in life and is what maintains life. And so we will hang on to energy any way we can. And so if we take in excess calories, we're storing them and we're going to save them for later in the form of fat.